Dear colleagues, dear friends, it's a great pleasure to welcome you to this new EHAT podcast. The podcast today will focus on the basic science of hemophilia gene therapy, a specific e-learning module that was recently presented by our colleague, Professor Paul Batty. So, Paul, it's a very great pleasure to welcome you today on this platform. So maybe you could introduce yourself.
Hi, thank you, Cedric, for the invitation to join today. So, hi, I'm Paul Batty. I'm a clinician scientist and I'm based at University College London and the Royal Free Hospitals in London. So I think my interest in gene therapy started a few years ago, and it really sort of ignited during my fellowship, which I spent a few years working with David Lillicrap in Canada, looking at sort of efficacy and safety in preclinical models. And what sort of led to this was seeing some of the data coming out of the phase 1 studies showing amazing results, and then wanting to explore this in more details, and then spending time in that lab, and then following this interest further, sort of coming back to the United Kingdom in a clinical role as well, so looking after patients who've had gene therapy going through the clinical studies. So thank you for inviting me to join today.
Well, thank you, and thank you to you for sharing your major enthusiasm and expertise. So, well, clearly, this is a podcast, so there will not be any visual, but you prepare very very nice slides and illustrations that are available on the EHAT platform. And what we would like to do today is to address a few important topics. The first one, some sort of general introduction to gene therapy, and then focus on the basic science of AAV vectors, because I think this is quite important, and then cover concisely important issues regarding efficacy and safety. So maybe starting with the basics of gene therapy, and when we discuss gene therapy, this is gene therapy for hemophilia. So what could you summarize? What are the current different approaches to make this gene therapy possible? I mean, the ingredients, the requirements, the strategies that are used. Could you elaborate a bit about this?
Yes, of course. So the term gene therapy is used quite broadly at the moment. So within the example of haemophilia, we have a monogenic disorder where there's a genetic defect. And the aims of the therapy is to provide a way that this can then provide a long-term treatment. So within the broad approaches to gene therapy, this can use either two approaches. One of these can be gene replacement therapy, and this involves the introduction of a functional copy of a gene into a cell to allow production of a normal protein. So that in this situation, you then have the genetic variant that's in the background, then by using this treatment can then allow normal protein to be produced. And this is the predominant form in haemophilia that's being used at the moment using AAV vectors, which we'll talk about today. The other approach, which is a little bit earlier in development, is looking at ways of trying to repair a defect in the genes. So with gene editing, or so more targeted at the site of a particular variant. And this is a little bit more in earlier development at the moment, but potentially is a way that some genetic disorders could be treated in the future for simpler variants within genes. So that's sort of the broad terms. And within any of these things, you need a cell to treat, you need a gene to introduce, and you need a vehicle for getting it into that cell in the first place. And with AAV, that acts as the sort of vehicle or vector for getting your normal copy of the factor VIII or factor IX gene into a target cell, which in our case is the cells in the liver, which are the hepatocytes.
Okay, so in terms of AAV, so what's the difference between a natural AAV and AAV that has been modified to perform gene therapy? So I think in your presentation you try to make a clear distinction between natural AAV and recombinant AAV.
Yeah, that's right. So the natural AAV virus is a really common virus. So it's something that many people listening to this podcast will have encountered over the course of their childhood. It's generally asymptomatic. And from a viral point of view, it's a very simple virus that needs other viruses present to replicate. And within its structure, which is a single strand of DNA, it really has sort of some elements that are purely there to allow it to sort of continue its existence. Within the virus itself, it's then coated in something we call a capsid, which acts as sort of an armor and also a way of directing it into a certain point. So when we use this as a vehicle, we take out the viral elements. So all of these sort of viral sequences that are there to help keeping this virus, it being a virus itself, are taken out. And in the position where this would be, so this is sort of cut out, we then replace these sequences with our gene that we want to replace, as well as a promoter or an enhancer element to try and direct this into a certain tissue. Additional things that we do as well is we can choose different capsids to try to direct the gene to the cell that we'd like to do. So we can use this as a way of being a vehicle, but also a vehicle that is more directed to one point. And for our case, we want the gene to go to the liver, because then that's where we want factor 8 or factor 9 to be produced. So within that final product, which is all made in the laboratory, we've removed the important genetic elements that makes that virus the virus, but keeping just the elements that keep the structure. So these are called inverted terminal repeats, or ITRs, and then sandwiched in this is the gene that we want to replace. So it acts as a neat way of directing the gene that you want to a site where you want to deliver the gene to.
Okay, so if we look at this AAV vector more closely, so there are very important components that you already addressed. So clearly the capsid outside, and then if we move inside, you will have the gene, the transgene, the promoter. So, in terms of capsid, do we have many different types of capsids?
Yeah, it's a good question. So within nature, there are many different types of AAV, and these are sort of divided in terms of how these have evolved. So there's at least 13 different AAVs in natural existence. These different capsids are there to help get AAV into a cell. So the different types of capsid are more targeted to some cells than others. And they use the different capsids will bind to different receptors on different cells. So some will be more specific to some than others. When we use capsids for gene therapy, depending on the therapy area where this is going to be used, different capsids may be selected. Sometimes we use natural capsids in that nature is always very good at doing these things, but there's also been various approaches to improve what nature does to try to avoid, say, immune responses and allow better uptake in cells as well. So it's sort of an important component, and also an area where some of the changes have been evolved in the next few years for gene therapy.
Okay, if we now move inside this capsid, so what we will do here is to replace the gene of the AAV gene by or gene of interest, so the transgene. So here, if I understand well, the promoter is important and also the sequence. So maybe a few words about the promoter or how important they are?
Yes, so the promoter is what gets everything started. So it's what gets production started when things are in the cell. You can have different sort of promoters. So some of these sort of can act in any specific areas, so they can be constitutive, and some of them are more specific. So in terms of trying to avoid some of the leakiness of a potential therapy, you can often use something which is directed towards certain cell type. And within all of the current vectors that are used for haemophilia treatment, these are very much liver-directed or liver-specific promoters to allow production in the liver cells where this arrives. So if there was a small amount in another tissue, you wouldn't expect this to allow a significant amount of expression. And we want this to be coming from the liver, because that's where factor 8 and factor 9 normally come from.
In your presentation, you shared with us a very few slides, but very nice about this concept of codon optimization. So for clinicians like me, this could appear as being quite complex. So what does that really mean, codon optimization?
Thank you, Cedric, for asking. So within the production of proteins in the body, you have amino acids, and within the genetic sequences for making up these amino acids, the DNA coding is based on 3 nucleotides. Fortunately, there is various different combinations that you can use for producing the same amino acid with these different combinations. So for some, there are multiple different sort of combinations of the three that you can use of the different nucleotides, and for other ones, it's more restricted. Some of these are easier for the cell to use than others. So some of them are quite rare and some of them are more common. So when you look at a sequence that you need within the cDNA for production of a protein, you can look at how these are used and how some of these may be more common, some of these might be more rare, and also potential implications that this could produce on the immune system as well. And that some of these repetitive elements in there could potentially be more immunogenic than others. So for optimizing, you're choosing things that really sort of try to increase the amount of protein that's produced at the end of this. And this is used in recombinant technology for production of protein. So it's not something that's new for gene therapy. It's used in many other areas. It's just using this in a more directed way when you're designing what your transgene is going to be to get your most production from that sequence.
So if I understand well, this is a process that is aiming at facilitating the production of this gene by the hepatocytes, for instance. So
yeah, that's absolutely right. So it's trying to get the most out of the gene that you're introducing from those hepatocytes and not making it more complex for them to produce than they need to.
And then there is another approach, and your slides are great. So this is, well, helping, the hepatocytes to produce better versions of the factor 9 or factor 8. So most of us are familiar with the factor 9 Padua, which is a super factor 9. But you also explained that for factor 8, there might be options to improve the factor 8 that's produced after gene therapy.
Yeah, so as you say, the factor 9 Padua has been a major advance for the treatment of haemophilia B, and this difference in this sequence is using a gain-of-function mutation. So within this, you're getting more activity than you might anticipate for the amount of protein that's produced. Unfortunately, there's not been something similar found for factor 8 as yet, which would be great if we could get that, because that may allow increased amount of factor 8 for a lower dose. But there's been various different sort of strategies which have been done to sort of aim for increased secretion and production. One of these, in terms of that's already going through clinical trials, has been using the V3 construct, which has been gone through the GO8 study. And in this, we've seen increased expression in mice following gene transfer. And that study's now been reported after many years of follow-up with expression in a number of individuals at a sort of more modest dose than is possibly seen in some of the other studies. There are numerous other approaches that are going through sort of the earlier stages, and these look at differences. So, for example, you might want to try and make it easier for the protein to be secreted, make it more stable, reduce the immunogenicity, also sort of affecting different cleavage sites in the protein to sort of just improve what you're getting at the end of it. It would be fantastic if we could find something similar to factor 9 there that gives that added benefit for the same amount, and maybe this will happen.
Yeah, certainly. Well, that's quite enthusiastic. But, well, and this is the last issue that you extensively covered in your presentation. This is the impact of immunity and immunology. So as hemophilia treaters, we know that the immune system has always been quite difficult with our patient, with inhibitors, antibodies. And this is also true when we discuss gene therapy. So clearly the immune system is involved. So maybe you could elaborate a little bit on the type of immune responses that can be observed during or after gene therapy.
Thanks, Cedric. The immune system is always important. So when thinking about gene therapy, I guess you've really got a few different phases. You've got the initial side of it is the baseline immune response. So is there an immune response already existing to a gene therapy vector? And for this part, we are often talking about whether there are antibodies to that vector, which could potentially mean that the vector is less likely to be taken up and effective. When you have sort of a treatment with any sort of novel stimulus, or whether that be a gene therapy vector or a protein concentrate, I guess there's a few different phases. You've got the immediate phase afterwards, which is a sort of reactionary phase that the body has, which is within the innate immune system. And then the secondary side, as things get a little bit more targeted, with either an antibody response being humoral or potentially a more T-cell driven innate response. And some of these areas have been looked at in different phases of sort of gene therapy, and each of them have potential importance. But as you said, the liver has been the key thing for discussion in the last few years.
Certainly. Yeah, so the liver reaction, this is something that we certainly fear after gene therapy. And what's in your view, well, what's your understanding of this? So what's happening in the liver when we see, you know, the liver enzymes going up after gene therapy?
Yeah, as you say, Cedric, this has been the big problem in the clinical studies, and we've seen sort of asymptomatic rises in liver enzymes early on after gene therapy, which in the initial studies heralded sort of loss of expression. We've now taken quite an aggressive approach in terms of immunosuppressing. There's been slightly variable findings in studies, but the current belief is that this is an adaptive response which is directed against the expressing cells. So looking at sort of something that's cytotoxic against cells, so loss of those cells afterwards, which has been seen in the initial studies, which was sort of delivered in the US and also between the US and the UK. But then there's also some uncertainty, and this response has also not been seen in some studies. So I think it's an area where there is a real need for active research into fully understanding. But our best understanding is this is relating to a sort of cytotoxic T cell response, which makes sense for targeting this with immunosuppression with things like corticosteroids around that time, because this has been shown in studies to allow ongoing expression through this sort of response in the majority of individuals.
Thank you. And well, maybe that would be one of my last question. You also emphasize the fact that clearly with gene therapy today, the gene that we introduce in the liver cells remain episomal. So for our colleagues who might be a little bit less familiar with this concept, so what does that really mean? And what are the current implications of this? And what would be the future? Will episomal status of these transgenes remains a reality in the future, or do you expect any change? A few words about this?
Yeah, so this is one of the big questions about how AAV works longer term, in that within a long-term format, the genome needs to form into a stable form. So it can either do this by two ways. One way is to form into what these terms that you said are called episomes. And within this, these are sort of circular structures, so they're round and they're containing double-stranded DNA. And within this format, these sit within the cell nucleus without binding into the person's genome. So they're sort of existing and allowing expression in a stable form that does not form part of a person's DNA. The other potential way this could happen is by sort of a process called integration, which is where a virus or other could introduce its DNA into a host, which in our case we're doing by virtue of a vector, and allowing that sequence to exist at some point within a person's DNA to allow expression longer term. Within looking at studies of AAV, the predominant belief is that most of these vectors survive in this first form, in these episomes, which is probably a good thing in that in this format they can exist and they don't have any potential implications on someone's DNA. Potential concern about this is as cells divide, we might wonder if these would get lost with cellular division, or if there are injuries to the liver for whatever reason, then when a cell's lost, then it would not be replaced. So that's sort of one of the potential worries. Integration is the other one, which it could be a good thing and it could be a bad thing, in that if you have integration into a safe area of the genome, and remembering that much of your genome is non-coding, this could then act as a stable way of allowing production of that transgene, say for factor 8 or factor 9, over a longer period of time. The worry is if that potentially could fall into an area which is important for coding and whether it has potential longer-term issues. From what we can understand so far, we know that episomes exist, and this has been shown in a number of studies now from humans, but integration does happen at small numbers. And there's an area we need to understand a little bit more about these processes. But from the studies in larger animal models and studies looking at integration today, we've seen no major safety signals in the laboratory so far.
Thank you. And for colleagues and friends who might have difficulties visualizing this, well, clearly your slides are excellent and clearly show this. So maybe, gene therapy is an area where you're certainly interested in and investing a lot of time and energy. So how do you see the future of gene therapy, let's say in the next 10 years? Any anticipation?
I mean, it's a good question. I think there's a lot of developments that are currently going through looking to try and generate that next generation of AAV vectors. I think there's also a lot of other forms of sort of gene and cell therapy, which are sort of at the earliest stages of development using different approaches. And I think it's going to be interesting to see which other vehicles are then potentially available in the future, whether this is using also targeted processes like CRISPR or other different vehicles like transposons and other things, which are being currently tried in studies. I mean, it's been great news that we've had approvals in many countries for some of the AAVs, and these really will be transformative for a number of individuals. But I think it really is watch this space, what the next forerunner is going to be. But I think it's an exciting time with a lot of developments currently going through early phase, so early preclinical studies, which will then hopefully come into the clinical trials in the future.
Well, clearly, all these prospects will certainly keep you a lot busy. So, Paul, I would like to thank you for contributing to this podcast. It was a great pleasure. I hope you enjoyed it, and I certainly hope that our audience enjoyed it, certainly in this fascinating area of gene therapy. So I would like to thank you again, and hope to have the opportunity to discuss again gene therapy with you in the future, Paul.
Thanks, Cedric, for inviting me to join today. Great to talk.